So welcome everybody to the July 2019 SNOC webinar, the Australian Inoculants Research Group. We worry about inoculant quality so you don't have to, by our own Jessica Rigg, and brought to you by the New South Wales Soils DPI Soils Unit. Now I'd like to introduce Dr. Jess Rigg, who's presenting today's webinar on the Australian Inoculants Research Group. Jess has worked with New South Wales DPI as part of the Australian Inoculant Research Group, or AIRG, since 2017. Microbial community analysis is where her research interests lie. Jess is particularly interested in how microbial communities can influence plant performance. Jess has worked with both beneficial microbes that promote plant growth, for example, rhizobia and arbuscular mycorrhizal fungi, and plant pathogens such as Phytophthora that cause disease. She investigated how microbial communities can influence the conservation of the critically endangered Wallamai pine. She's worked with the Royal Botanic Gardens in Sydney to study the impact of the root rot pathogen Phytophthora on alpine plant species. And she was involved in work on how amendments used in the current New South Wales DPI GRDC subsoil constraints project affected microbial communities. Now, I know you're dying to hear from Jess.
Okay, good morning. So it's really excellent to hear that some of you have heard about the green tick, and if you haven't, I'm going to be able to tell you about it today. So there's so many factors to consider when you're planting a legume, and so our group want to ensure that rhizobial inoculants that have our green tick are the best quality they can be, so that it's one thing farmers actually don't have to worry about is inoculant quality. So on the screen are some examples of some beautifully nodulated plants and some of the impacts that inoculation can have and the benefits it can have to legumes. So I wanted to start at the very basic beginning of how a nodule is formed. So when a seed germinates, as the roots grow, the plant releases flavonoids and other compounds, alerting the rhizobia to their presence in the soil. So the rhizobia respond to these plant signals by releasing their own compounds, and these are controlled by all sorts of nodulation genes. So as this molecular dialogue begins, the bacteria the root hair starts to form around the nodule. So this molecular dialogue is highly specific, and it's specific to the bacterial strain and the plant species and cultivar. So as this connection continues, the root hair curls around the rhizobia and an infection thread forms down into the root. A bacteroid or nodule forms. So next is a really example of a well-nodulated bean plant. So you can see a large number of nodules in the crown of the roots, and they're pink, meaning that they contain a plant-produced compound called legume, which is extremely important. It's essential to that, fixing nitrogen from the atmosphere into the soil. So rhizobia have a highly plastic genome. So basically, rhizobia can actually lose their ability to nodulate a host. So within a bacterial cell, there's the bacterial genome, which is a large circular chromosome. And if you'll think about this in terms of a computer, the chromosome is like the computer's operating system. It controls the day-to-day functioning of the bacteria. So bacteria also contain plasmids, small circular pieces of DNA. So these you can think about like software or apps. So basically, on your computer, you can delete a program or an app, and you lose the ability to perform the function that this app contains. So in the same way, nodulation genes are often included on plasmids of bacteria. And bacteria can actually lose these plasmids, or they can swap the information carried on these plasmids with other bacteria. So essentially, for some of our rhizobial strains, they may be the correct bacteria, but they can lose the ability to nodulate a host. So some of the forces acting on rhizobial evolution when they're out in the field include stress and competition. So basically, when an elite rhizobial strain is in the soil, it's competing with the whole soil microbial community. It's competing for space and resources. So when the rhizobia is free living in the soil. It's living as a saprotroph. So the genes that it contains to nodulate a host are expensive to maintain. So the costs to maintain the symbiosis genes are high, which means that they're more likely to lose these genes. So this is why, for our group, it's so important that we test our elite commercial strains annually to check that they still have this ability to nodulate the correct host, and we take steps to minimize genetic drift. So why do we even care about all of this? So this is quite complex to start. I'm going to talk about the benefit of inoculation. So there's many, many benefits to planting legumes and for them to be well nodulated. So you get increased plant biomass and forage quality, you can get increased grain yields and carryover for subsequent crops, and they can provide a disease break in crop rotations, plus many other benefits I haven't mentioned here. And this slide actually shows just a concentration gradient of high inoculant concentration to low, and you can see the benefit that inoculation gives. So in 2012, there was a back of envelope calculation that was done that said if all the area sown to legumes in Australia were fixing nitrogen optimally, we'd be saving $4 billion of nitrogen fertiliser annually in fixed nitrogen, which is a huge benchmark to live up to. So now I want to introduce my team, the Australian Inoculants Research Team. So we're a small soil microbial team based at Elizabeth MacArthur Ag Institute at Menangle. And so the role of our group is to basically ensure that Australian farmers have access to high quality microbial products. We facilitate a robust independent quality control system for microbial products. So we independently test products for inoculant manufacturers. We hope to increase consumer confidence in inoculant products, so by making sure they contain the right thing in the right numbers. And we also conduct soil microbial research. So our group curates the National Code of Practice and Green Tick Scheme, which I want to tell you about today. So basically there's two parts to this National Code of Practice. There's the protocols for selecting the elite commercial strains in Australia, and there is also the current numerical standards for legume inoculants. So under this program, there are 41 official commercial strains that have been developed over many, many years, and these cover over 100 legume species. And then within these species, there's many, many legume cultivars. So there's quite a broad reach of these commercial strains. So our National Code of Practice and Green Tick program are voluntary opt-in programs. So basically we have 4 major industry partners, which are listed there on the screen. And these partners choose to get their products independently tested by our service. And then when we independently test them, they are allowed to show the green tick logo on their packets. So if you see an inoculant bag that has the green tick logo, you know that it's been independently tested by us. And some of the things we're looking for is that the labeling on the package is correct. It shows what strain of bacteria is in the bag. We check that the correct rhizobial strain is there in present numbers and it meets in sufficient numbers, so it meets that quality standard. We also check that the product is free from contaminants. Contaminants in a batch may grow and outcompete the rhizobia in the packet, reducing rhizobial numbers and hence increasing the risk of low nodulation. So we make sure that there are low contaminants. And we also check that the product, after being manufactured, still nodulates a host. So when all of these things have been checked, we issue a green tick. So basically, if you purchase a product with a green tick, you can be assured that it's gone through that quality, those quality steps. So another key role of our group is to supply the commercial mother culture strains to industry. So we do annual mother culture testing. And as I mentioned, there's 41 commercial strains. So basically each year we check that the strains are pure, that the identity of the strain is correct, that strains can scale up for production. So basically they can grow to a large number of organisms, which is important for manufacture, and making sure those strains haven't lost those genetic essential genetic components required to form nodules. So an example on the photo here on the screen is of effectiveness testing. So basically, it's a subsample of an experiment where you can see plants that are inoculated are much larger and healthier and producing nodules than plants that weren't inoculated. So we also take these, we use a parent lot, working lot system. So basically, we're trying to minimize the amount that these bacteria are subcultures on and on and on to minimize genetic drift. So all the cultures are kept freeze-dried in as close to a single generation as we can, so we aren't changing these bacteria as the years progress. So it's really amazing. The Australian rhizobial inoculant market is renowned for being a really high quality and it sets a high standard worldwide. And this is in part because of this quality assurance program and because of the steps taken, the extensive steps taken to select a strain. Australia is unique in that we have one strain for one host group. So instead of every company or every research group having their own strains for commercial use for different legume hosts, there is one strain for one host group in Australia. And the process to change this strain for a host group is quite extensive. So I wanted to run you through some fun facts about some of our strains in the commercial program. So the chickpea strain, CC1192, has been in use since 1977. The strain was originally isolated from Israel, and it's a Mesorhizobium ciceri. Some other fun facts that I wanted to point out are that despite some of the strains being very taxonomically similar, so for example, the Rhizobium legumisaurus has two biovars, and they, despite being taxonomically similar, they nodulate very different host groups. So the perennial clovers and annual clovers have different strains to the field pea, vetch, faba bean, and lentil group, despite it being the same, almost the same bacteria taxonomically. I also wanted to mention sometimes you have a very similar bacteria that can't cross-nodulate. So a good example is the chickpea and bicerula strain. They can't nodulate. So the bicerula strain can't nodulate chickpea and vice versa. So there's some very cool, neat, specific interactions going on. So now I wanted to talk about how strains are selected for this elite commercial program. So they have to be effective and they have to have the appropriate host range. So the manufacturers, and I assume farmers, wouldn't want to be buying all these different strains and having all these different inoculant types. So basically, we want to have a reasonably broad host range and we need them to be effective. So the strains need to be as genetically stable as we're able, so not a strain that's likely to lose those genes for nodulation quickly. The strain needs to be able to be stored and can easily scale up for production. So some strains can be amazing at fixing nitrogen, but will not grow up to sufficient numbers that we need them to be able to manufacture them. Another thing that we check for is their ability to survive in a carrier like peat or on seed. So if you were to go about the process of changing a commercial strain, for a major group, so a major group is something like chickpea or the faba lentil group, you'd need 10 field trials over a minimum of 2 seasons. And in these field trials, you'd gather data such as the new strain versus the current strain versus an uninoculated plot. The product would be applied, the strain would be applied as peat, and we measure all sorts of things like nodule score, nodule weight, herbage yield, and grain yield. So once all of this research done, the report's prepared by the person researching that strain. Then the strain goes under an independent assessment by our group, and then AIG facilitates a vote, and it's put to key members of industry, and they vote in the strain or not, depending on that data. So I wanted to run through a case study now. So Desmanthus is a strain. Desmanthus is a tropical legume species. So in 2017, we discovered that the strain CB3126 for Desmanthus was no longer effective. So it wasn't producing nodules and it wasn't fixing nitrogen with Desmanthus. So we withheld this strain from the market, the inoculant market, and in collaboration with the Centre for Rhizobium Studies in Murdoch Uni, we recovered an effective strain of the Desmanthus strain. And then this was reissued to inoculant manufacturers. And it was a good example of why our quality program is so important. So had this not been noticed, there would have been an inoculant that was produced that wasn't effective. So it was critical that we had checked this, had discovered it was not nodulating, and then took appropriate steps. So that's one example of where our inoculant checking program has been useful. So now I wanted to talk about the current state of play. This is not an exhaustive list, but some developments needed for the Australian rhizobial market, in particular with a flavour of what our group is focusing on at the moment. So soil constraints, particularly soil acidity and soil acidicity, are limiting where you can plant high-value legumes, like some of the pulse crops. So there's work going on around developing acid-tolerant strains, and in particular, this works further along for the pea, faba, lentil strain and also the chickpea strain. There's also a lot of interest around developing innovative inoculant delivery technology that overcome some of these soil constraints. One thing that's also critical is that we keep this plant rhizobia specificity that I was mentioning earlier. So we keep checking strains against their cultivars. So new cultivars of plants are being bred all the time, and it's really important we check these new cultivars can still nodulate with the commercial strains. So every new legume cultivar should be checked. And also the development of farmer decision support tools. So tools to determine what strains are present in my nodules or soil, and do I need to inoculate? So the first project that our team is involved in that I'll talk about is a GRDC Pulse Rhizobia project. And basically, it's all about increasing the effectiveness of nitrogen fixation in pulses through improving rhizobial strains. So this is a major body of work in this a major component of this project is the commercial strain replacement of group E and F. So that's currently WSM 1455 or SU 303 for faba beans, lentils, pea and vetch. So 4 new acid tolerant strains have been shortlisted from a large amount of work. 2 of them come from Western Australia, from Ron Yates's group, and 2 of them come from Ross Ballard's group at Sardi in South Australia. So there's a number of field trials going on to assess these strains, and EGG, our group, will be performing experiments testing these strains to tolerance to herbicides, herbicide residues, and also to pH. We're also checking whether they can be manufactured and that strains are stable. So the aim is by June 22 to have improved rhizobial strains for lentil, faba beans, and also for chickpea. So another project that we've got going on is the High Rainfall Legumes project. So basically this project is about broadening the zone of adaptations of legumes in the high rainfall zone by better soil management. And the whole aim is to address total feed base problems and also feed gap problems using legumes. So basically we've got field trials assessing around 20 legume cultivars running from Victoria all the way up to the top of New South Wales in that high rainfall zone. So this is a broader project being run with the New South Wales Department of Primary Industries Pastures team, and it's co-funded by Meat and Livestock Australia. So our group's part in this project is to look at host compatibility. So when you're sowing all these legumes in a mix, which strains they're associating with out there in the field, and looking at rhizobia and legume persistence and nodule occupancy. So this next figure that I'm going to show you is quite complex, but I'll tell you the key message that I'd like you to take away from it. So if you look at the white clover, so this is white clover inoculated with both the recommended commercial strain and then the strains of other pasture species that are likely to be sown in a mix. So, for example, with the clover, it will form nodules with the recommended commercial strain, and that is the most effective. But it will also produce nodules that are less effective with many other strains, many of the other commercial strains. So this sort of host compatibility becomes one of the reasons why you may see variations in nitrogen fixation in the field. So far, we're suggesting that you select pasture mixes that have similar rhizobial needs, or if you're going to sow multiple hosts together, to ensure that legume seeds are inoculated with the most effective rhizobial strains separately prior to sowing. So moving on, the next part of work that I'll talk about is our involvement in the DPI GRDC chickpea bilateral. So this project is around improving the management of Phytophthora root rot of chickpeas. So briefly, I'm going to talk about some work that was done out of Western Sydney University. So basically, Jonathan Plett and his colleagues noticed that when you're breeding Phytophthora-resistant chickpea, you're breeding out the ability of those chickpeas to talk to the pathogen, the Phytophthora. And he showed that the molecular dialogue that I was talking about at the beginning of this talk between the pathogen and the plant is similar to between rhizobia and the plant. So Phytophthora-resistant chickpea lines produced better nodules than Phytophthora-resistant lines because the ability to talk to that rhizobia had sort of been selected away from. So in this project, this improving management, the chickpea bilateral project, they're breeding new Phytophthora-resistant lines of chickpea, and AIG's role will be to review the nodulation capacity of the shortlisted Phytophthora-resistant chickpea cultivars, with the aim of choosing a chickpea cultivar that is both Phytophthora-resistant and able to produce really robust nodulation and nitrogen fixation. So the next thing I'll talk about that our group is working on is a project around developing a real-time decision support tool for legume growers. So Ashley Webster, one of the technical officers in our group, won the Minister's Award for Science and Innovation. And basically, her project will sequence the full genome of 21 of the commercial rhizobial strains. And Ashley will be using iron-tolerant sequencing, and this is a tiny little sequencing platform, as you can see in Ashley's hand, and we'll generate from these genomes, we aim to generate a fast, portable, accurate, and cost-effective method of identifying rhizobial strains. These will be real-time molecular assays that we hope to be able to use in the field. And basically, the overall goal is to develop a farmer decision support tool, whether or not they need to inoculate. So with this, I'm going to end and ask if anyone had any questions on what our group is doing or about the green tick. So thanks for listening.
Thanks, Jess. Very concise and informative. I'll just give people a couple of minutes to get their questions ready. Now, Vicky Brun has got a question. So Vicky's asked, I'm not sure what products the green tick is put on. Is it specifically rhizobial products?
Yes. Okay, so Vicky, yes, it is specifically rhizobial products. We work with BASF, New Edge microbials, green microbes, and Novozymes. So it's usually on peat products, and we're working towards a protocol to test granules as well. So look out for it on peat rhizobial products at this stage. So there are liquid rhizobial inoculants. There tend not to be fertilizers that include rhizobia. So rhizobia are obviously living organisms, and some of the components in fertilizer, like copper and zinc, are really toxic to rhizobia. So one thing we say is to minimize the exposure of rhizobia to fertilizers. There are non-rhizobial microbial products on the market. They're often mixtures. Our group doesn't test those.
We've got Joe Powell's. The question was, can Jess expand on the producer tools that you mentioned that are being developed?
Yeah, sure. So basically, we are sequencing the genomes of the commercial rhizobial strains with the hope of finding a region and developing a molecular assay. So we have a portable, real-time quantitative PCR machine that we can take into the field and that can use solar. So we're hoping to be able to tell farmers what strain is in their nodules. So sometimes we have the issue of a crop has, like a legume crop hasn't been inoculated and there's ineffective white or green nodules on the plant that aren't fixing nitrogen. We can say, we can sort of give them an idea of the need to inoculate, what's going on inside the nodules. So that's the plan. This project's just kicking off. So we received the funding for this month. And so all that sequencing is going to begin shortly.
Terrific. And next question's coming from David Rickards. So David's question is there a reasonable chance that soil acidities of 4.5, pH 4.5, will be able to sustain nitrogen fixation?
Oh, okay. It could be borderline, but that's what we're assessing in this current project. So we're going to check the tolerance of these new acid-tolerant rhizobia to pH 4.5. And we're also, there's a number of field trials going on to check these new rhizobia and whether they can survive in these sorts of really acidic locations. So stay on the lookout for that information, but it'll be coming soon. We hope to. It would be ideal, but we are working with living organisms, so stretching that tolerance may be difficult.
Thanks, Jess. So now Lachlan Ingram's got the next question. Here you go, Lachlan.
Yeah. So I was just wondering what the current state of play is in regards to the effectiveness of freeze-dried inoculants.
Yes, freeze-dried inoculants contain, they're very effective. So they contain a very large number of organisms, more than peat, and that means you have to apply less of them to get the desired target number of organisms. So our product, our green tick, does go on freeze-dried products from New Edge Microbials, and they seem to be of a great quality.
Okay, cheers. I'm just also wondering, what is the number of the 4 companies you're working with? What sort of proportion of the market do they sort of, I guess, have access to or control?
I think that'd be a question more for them than us. Yeah. I'm not sure what their market share or proportion is, and I couldn't really comment anyway.
Okay. I was just sort of more wondering, those 4 companies, are they sort of the predominant producers, or are there potentially a lot of other producers which are not getting green tick, which are available to farmers to access?
Sure. So they do cover the major companies that produce rhizobial inoculants. So they don't cover non-rhizobial inoculants, but the green tick is really tailored around rhizobial products. So I think we do capture the main players in the Australian space. Okay. Aside from some of the people producing granules in Western Australia, perhaps.
Okay, cheers, thank you.
Thanks, Lachlan. So now, people are being very patient. We've got Chatarika Dalagala. I hope I've got it right, Chatarika, so I'll read your question out. So Chatarika says, nice presentation, Jess, and so to the work of your group. I have one question, whether you guys test the effectiveness of co-inoculation of inoculants when inoculated with the potential resident strains you find in the field, because in general, residents are inefficient in nitrogen fixation, but are competitive and resistant in field conditions.
Yes, thank you, Chatu. That's an awesome question. So at present, our work hasn't included testing co-inoculation with that, but there is a large body of work focusing on that, because you're right. When you inoculate the elite commercial strain into the field, it's competing with all those background rhizobial populations that are there, that are present in the field. A good example is some of the work coming out of Western Australia working on the Bicerula strain. So when Bicerula was introduced, there was no history of that rhizobial strain in the soil. And as that rhizobial strain became resident in the soil, it swapped its genes with some of those background rhizobia that were sitting there. So some of those background rhizobia gained the ability to nodulate with Bicerula, but these nodules that they formed were ineffective, so they weren't very good at fixing nitrogen. So basically going forward, there's some real interest in developing strains that won't give their genes to those resident, like stabilizing, making sure they're genetically stable. So yes, there's definitely work going on out there in the rhizobial inoculant field about background strains. Some of our work in the high rainfall zone project is actually getting at that sort of the influence of background populations on the effectiveness of inoculation. So we're happy to chat to you more about that if it's something that interests you.
Terrific. Thanks, Jess. Thanks, Chaturika. So that's a quick one. It might be a dumb one, Jess. You started your talk by saying how specific the interaction between a rhizobial strain and their host legume is. Yet I noticed some of the strains, for example, Group E, could be used for many hosts, so field peas, vetch, faba beans and lentils. How can that be?
Okay, yeah, that's really true. So the plant and or rhizobia may be either a generalist or a specialist. So some legumes like cilantro have a huge, broad range. So they'll nodulate with many different rhizobial species. An excellent example of a very specific relationship is the case of the Mesorhizobium ciceri strain for chickpeas. So chickpeas will only nodulate with that strain. And so, yes, it comes down to whether the plant host and or bacteria are either a generalist or a specialist. So it kind of really complicates the relationship between strains and plant species. And also, I'll add, it's one reason to consider the background populations of rhizobia at sites where you're going to plant a legume, because you may end up planting a legume that will nodulate with these background populations and not be as effective at fixing nitrogen. So history of inoculation is important in paddocks.
Okay, terrific. I just thought you had a really good question here that you got in orange this week that you let me know about, and that was, I think if you could mind covering that for a second, how will I know if I need to inoculate?
Sure. So the first thing I'm going to say is that the relative cost to inoculate is really low, especially in cases where the loss associated with nodulation failure could be high, for example, in a pulse crop like chickpeas or lentils. So basically, it's best practice just to inoculate when you're planting legumes, regardless of if you think they need it or not. So you'll probably see the greatest positive response to inoculation where a paddock hasn't had a history of that legume or hasn't had a recent history of that legume being grown. And it's important to remember that even in a paddock where you've recently grown a legume, it's not true that all soils are best for the survival of rhizobia. So you may need to inoculate again. So basically, rhizobia may struggle to survive in some soils where they're super acidic or they have a low clay content. So it's difficult at the moment without tools to detect that are commercially available to detect rhizobia. So I'd say if you can inoculate as best practice, yes.
Thanks, Jess. I'm just making sure I've got the right reference there. JDC booklet inoculating legumes. Yeah. So that should be, hopefully you can see that all right on your screen, Harry.
Worst case scenario, you can just Google inoculating legumes, a practical
Thanks,
guide.
Jess. Okay, I'll leave that up on there. Just Yeah, so well done, Jess. Thanks. Sorry for. I know you've got a busy thing to rush off to, so thanks for taking the time, and thanks everybody for tuning in today. And we'll look forward to seeing you at next month's webinar. So thanks again, Jess.
Thanks, Luke.